We'll be reviewing page 26, which is about DNA versus RNA. Remember that the process of protein synthesis means we have to take DNA and we need to make proteins out of it. But in order to do that, we have to have the molecule RNA. And today, in this video, we're going to talk about what is RNA. So, down here, there are three differences between DNA and RNA. DNA, you guys already know that the sugar found in the center of DNA nucleotides is deoxyribose. You also know that DNA is a double helix. And you know that DNA has the nitrogen bases A, G, C, and T. Now, in RNA molecules, we have almost the same situation, but there's three main differences. They're still made up of nucleotides. Okay, they're both types of nucleic acids, which is why DNA, RNA, the NA stands for nucleic acids. So they are both the macromolecule nucleic acid. But that D and R at the beginning tells us that the sugar is different. So D for deoxyribose, R for ribose. So this molecule has one less oxygen, deoxy, okay, than ribose does. So this sugar has an oxygen in it that this one doesn't. That's a difference between DNA and RNA. The second difference is that RNA is single-stranded. It doesn't form a double helix. It stays as single strands. The third difference is that we don't have T's in RNA. Instead, we have U for uracil. So that is the nitrogen base. We still have A, G, and C. And everything does normal base pairing, except we're just replacing the T with a U. So A and U will pair together instead. C and G still pair together in the same way. Now, what are the different types of RNA? So now that we know what RNA is, RNA can be made into a copy of DNA. Now, there are three main types of RNA. There is messenger RNA, which we say mRNA is what it stands for. We say transfer RNA is known as tRNA, and ribosomal RNA is known as rRNA. Now, messenger RNA is a copy of a gene. So it contains the code that DNA has. Every three letters together makes up a codon, which can be coded for by our amino acid chart. Like it says here, it's a disposable copy of a gene that DNA that can leave the nucleus and travel to the cytoplasm. This type of RNA is read 3 letters at a time. Each of the 3 letters is referred to as a codon. That's important because tRNA, known as transfer RNA, helps transfer an amino acid to the correct spot in line to make the correct protein. So this 3 bases is called an anticodon because it should match up with the codon. So UAC matches with AUG, for example, lining up a methionine as the first amino acid in the protein chain. The very last type of RNA is not one that has a specific code. It doesn't have codons or anticodons. These little squiggles in here are the rRNA. And rRNA literally just makes up part of the physical ribosome. So it's helping up making up this structure of this ribosome. That's important because the ribosome is what holds the mRNA and the tRNA together and then helps make the bond between each amino acid. So we'll learn about that later. Thank you. Bye-bye.